Okay, thank you, Amanda, for that introduction. And thank you everyone for joining us today. I'm really excited to tell you more about this topic and tell you exactly how Stem Cell Technologies as a company is trying to approach the challenge of editing human pluripotent stem cells with high efficiency and to obtain high quality edited cells. Okay, so I'd like to begin by just noting that genome editing as a field has really been around for quite some time. Only recently has it been propelled into mainstream use, and this is particularly due to the CRISPR-Cas9 system and its increased feasibility over more traditional genome editing methods. So instead of relying on protein DNA recognition, like these more traditional methods, meganucleases, zinc finger nucleases, and TALENs, the CRISPR-Cas9 system, or the Cas effector nuclease, is based, or targeting of this nuclease is actually based on Watson-Crick base pairing between a guide RNA molecule and the target genome. And so the simplicity of guide RNA design of new targets and the need for only a single constant protein, so this Cas effector nuclease, instead of having to engineer unique proteins for every target site, you only use one protein. So this decreases the cost and complexity of an experimental design. These features have really made the CRISPR-Cas9 platform accessible to researchers around the world. So I'd just like to highlight that regardless of the technique, all genome editing relies on the formation of DNA damage. And so for the most part, with these technologies, you target a specific site in the genome and generate a DNA double-stranded break by these nucleases. And so this DNA double-stranded break will then be repaired by endogenous cellular repair pathways. And it's really our ability to predict or coerce the cell into using a specific pathway that dictates genome editing outcomes. So I'm just briefly going to go through the mechanism of how CRISPR-Cas9 works. And today in my talk, I'm going to focus on the Cas9, Cas effector nuclease. So as I mentioned, there's the Cas effector protein, or Cas9, which is shown in this bean shape here. And the other part of the system is this guide RNA shown in blue. So traditionally, the guide RNA is made up of the CRISPR RNA and tracer RNA molecules, which interact at the 3' end of the CRISPR RNA to generate a scaffold for interaction or binding with the Cas9 protein. Probably the most important part of the system, or at least of the guide RNA, is this 5' end of the CRISPR RNA, which is also known as the protospacer or targeting sequence. And so this is a 17 to 24 nucleotide sequence, often 20 nucleotides, that's complementary to the target region of the genome that you want to edit. So once Cas9 pairs with its target guide RNA, it'll scan the genome for something known as the PAM site, or protospacer adjacent motif. So for Cas9, this is NGG, where N is any nucleotide followed by 2 guanines. And when it finds a site that also immediately upstream is complementary to the target region of the guide RNA, Cas9 will unwind the DNA and generate a DNA double-stranded break exactly 3 to 4 nucleotides upstream of that PAM site. So as I mentioned, after that double-stranded break occurs, there's then endogenous cellular DNA repair pathways will take over. And there's 2 main pathways of DNA double-stranded break repair in mammalian cells, and that's non-homologous end joining, or NHEJ, and homology-directed repair, or HDR. And so the non-homologous end joining is the predominant pathway that's active in most cells, and typically cells will choose to undergo this pathway. The way it works is when you have a double-stranded break, there's direct ligation of the broken DNA ends back together. Now, often this does not result in any mutations forming, and so there's high fidelity of repair using this system. But in the context of CRISPR-Cas9, if the sequence is repaired back to wild type, then Cas9 can rebind with the guide RNA and generate another cut, and this continues until imprecise processing can lead to disruption of either the target binding region or protospacer region or the PAM site. And when this occurs, indels can form, and this can be very useful for a number of different types of studies, such as studying functional gene knockouts. So if you generate or target CRISPR-Cas9 to the coding region of a gene and generate an indel that causes a frameshift mutation, this can allow you to study the consequence of genetic knockout of your gene of interest. Alternatively, you can also use this strategy to delete regulatory elements. So if you target CRISPR-Cas9 to, for instance, an enhancer element, indels can disrupt binding of proteins to that enhancer element and allow you to study those types of regulatory elements. The alternative pathway is homology-directed repair. And this pathway actually occurs at quite a low frequency, and this is because it's most active in late S or G2 phase of the cell cycle after the cells have replicated their DNA. And this is because under normal physiological circumstances, the cell, if there's a double-stranded break after replication, it'll utilize the homologous sister chromatid as a template for repair. In the context of CRISPR, the user can introduce exogenous donor DNA that can act as a template for precise knock-in of small point mutations or even large gene insertions. And so that knock-in template will look something like this, where you have homology arms spanning the break site and whichever or whatever type of change you want to make, shown in blue, that will be precisely encoded into the genome. And so this pathway is quite powerful, but as I mentioned, since it exists at a pretty low frequency, it's quite challenging to achieve this type of editing with high efficiency. But it does enable precise rewriting of the genome, which is really powerful, especially in induced pluripotent stem cells for disease modeling applications, where you can correct or introduce patient-specific mutations in a cell line to study the consequence of those mutations. Alternatively, you can also take cells from a monogenic disorder, repair that mutation, and then introduce them back into patients. And this strategy can potentially be used to repair genetic disorders. So when you're applying CRISPR-Cas9 to a cell culture system, you have a number of different choices in the cell culture model that you're going to work with. So in the early days of using CRISPR-Cas9 in mammalian cells, traditionally people used immortalized cell lines. So these are cell lines that have acquired a non-physiological ability to proliferate indefinitely in culture. And this is because they're often isolated from tumors, or they can be immortalized with viruses. And these cell lines are very useful because they can proliferate indefinitely in culture. They're very easy to maintain and propagate, and often quite easy to manipulate as well. However, there's some drawbacks with these cell lines in that they do not very closely mimic in vivo conditions, and they harbor several chromosomal aberrations. And this is because they come from typically a background of tumorigenesis. And so some examples include HeLa, A549, or HEC293 cells, which are very powerful cellular models, especially for studying basic biochemistry. However, when you want to get into more physiologically relevant models and disease modeling, there's some other cellular options that might be a better fit. And these include primary cells and induced pluripotent stem cells or embryonic stem cells. And at STEMCELL, we collectively refer to these two cell types as human pluripotent stem cells, or HPSCs. And so how these differ from immortalized cell lines is that both of these cell types are isolated from an individual donor. So they are of human origin, and they're taken right from the patient and can be put into culture and either cultured immediately like primary cells. So in the case of CD3 positive T cells or CD34 positive hematopoietic stem and progenitor cells, these can be isolated from blood and put directly into culture. Or with iPS and ES cells, you can, or I guess iPS cells at least, you can take patient cells like skin or blood or other various cell types and reprogram those cells into a stem cell-like state. So both of these types of cellular systems more closely mimic in vivo conditions, but where they differ is in their ability to divide in vitro. So primary cells have a limited cellular lifespan in vitro, and over time can lose some of the characteristics that you require of them, such as potency of the stem cell fate. However, the power, or I guess why HPSCs are such a powerful and attractive model is that they can theoretically divide indefinitely in vitro. Also, both of these cell types exhibit minimal chromosomal aberrations because they are kind of more directly coming from a patient. I'm going to talk a little bit later about some chromosomal abnormalities that can occur with long-term passage of PSCs and why it's important to be aware of these. But overall, the level of chromosomal aberrations is quite low in these cell types compared to immortalized cell lines. So with HPSCs, some examples are that thousands of IPS lines have been generated and are currently being generated by reprogramming patient or healthy control cells into IPS lines. And really, again, where the power comes in is that these stem cells can be then differentiated using robust protocols that have been described in the literature for decades now into really almost any cell type in 2-dimension or 3-dimension. And these are especially important when, in the case of primary cells, you can't have access to that kind of tissue, like with cardiac or neurological tissue. With IPS cells, you can differentiate those stem cells into those kinds of, I guess, tissues or cell culture models that are not readily accessible. Okay, so despite all of this excitement, especially when you can apply genome editing to these pluripotent stem cells, you can then generate physiologically relevant custom human cell culture models. However, there are still some challenges that remain when editing HPSCs. And so throughout the presentation, I'm going to delve into some of these challenges and how we as a company are trying to address them at STEMCELL Technologies. So the first challenge I'm going to describe is the difficulty in expressing CRISPR components in the cells due to cytotoxicity or degradation of the components. Then I'm going to get into how HPSCs are inherently more challenging to culture and manipulate than immortalized cell lines, and some ways that we have tried to enhance the culture experience for these cells so that they can be easily manipulated. Another challenge, which is a very significant challenge, especially when editing these types of cells, is that they exhibit low clonogenicity and low single cell survival. And so the derivation of clonal cell populations can be quite challenging. And then finally, as I mentioned earlier, all genome editing relies on the formation of DNA damage. And it's known that pluripotent stem cells are quite sensitive to DNA double-stranded breaks. And so the DNA damage response in these cells can constrain editing efficiency. So I'll discuss this and potentially ways of monitoring genetic stability and trying to overcome this challenge. Okay, so first, I'd just like to introduce the different methods of expressing and delivering the CRISPR components into the cell. So this is a schematic of a cell. The outside is the cell membrane, and the inner circle is the nuclear membrane. And there's three main techniques that people use in order to deliver CRISPR components into the cell. So the first includes expression plasmids. So you can encode the Cas9 gene alongside your custom guide RNA on a DNA plasmid, introduce that plasmid into the cell, which will then be transcribed, translated into the Cas9 protein and guide RNA, which can then form a complex known as the ribonucleoprotein complex or the CRISPR-Cas9 complex to facilitate genome editing. Alternatively, Cas9 mRNA can be introduced into the cell alongside synthetic guide RNAs or in vitro transcribed guide RNAs. And then the mRNA will be translated and then go on to form the ribonucleoprotein complex. And then finally, the last method is pre-complexing of purified Cas9 protein with synthetic modified guide RNAs and direct delivery of that RNP complex into the cell. And so this is the system that we offer here at STEMCELL Technologies. And we feel that it is the best system for use with complex cell types like primary and human pluripotent stem cells. And this is because it's the most direct method of delivery of the CRISPR-Cas9 complex. As soon as it enters the nucleus, it's ready to be functional. And there's also, it overcomes the cytotoxicity associated with introducing foreign nucleic acids into the cell. There's also a few additional features that I'm going to go through in the next slide. So this is some data showing benefits of the RNP system over DNA or mRNA-based systems. So the first is that it exhibits enhanced performance across a number of different electroporation conditions in human cells. You can see that the protein or red bar often or always exhibits higher indel production or editing efficiency compared to DNA or mRNA-based formats. Probably the most key piece of data related to this is shown in the middle panel here. So this is a Cas9 Western blot, and cells were taken over a time course after delivery of CRISPR-Cas9 components using either DNA, mRNA, or protein. And so first you can probably notice that the mRNA-based expression system exhibits quite low expression of Cas9. And this is in comparison to RNP methods, which peak at around 4 hours, exhibit quite high expression of the Cas9 protein, and then this is reduced over time, and by 72 hours, there's a complete lack of Cas9 protein expression within the cell. This is in stark contrast to the pattern that you observe when you introduce Cas9 or CRISPR-Cas9 on expression plasmids. So here, peak expression of Cas9 is observed after about 24 hours, and it's maintained or prolonged up to 72 hours and potentially even longer after delivery. And so this indicates that Cas9 is kind of hanging around the cell for an extended period of time, and this could potentially result in a higher off-target effects. And this, I guess, observation is nicely reflected in the data on the right-hand panel, which measured off to on-target ratio comparing DNA, RNA, and protein systems at 2 different off-target sites, showing that RNP-based methods showed the lowest off-target effect compared especially to the plasmid-based system. So even when you're, I guess, there's a choice of how to express Cas9, but there's also a choice of how to deliver the guide RNA in your cells, and this can also greatly impact editing efficiency. So as I mentioned, you can encode your guide RNA on plasmid DNA alongside the Cas9 gene. You can also deliver the guide RNA as in vitro transcribed guide RNA that you purify in the lab, or you can purchase synthetic modified guide RNAs that contain these chemical modifications on the ends that are depicted here with red asterisks, typically 2'-O-methyl and phosphorothioate linkages on the ends of these guide RNAs can help prevent nuclease degradation. And this is especially important in cell types like immune cells that naturally want to degrade any kind of foreign RNA that comes into the system. And so at STEMCELL, we offer these synthetic modified guide RNAs, and we feel that they're the best system to work with purified Cas9 protein because they exhibit very high flexibility in terms of the cell types that they are effective in. So they work in cell lines, but they also work very well in stem and primary cells. And this is in contrast to plasmid DNA and in vitro transcribed single guide RNA, which are most effective in cell lines and can show efficacy in stem and primary cells, but their efficiency is usually variable or at least lower than the RNP-based system with synthetic modified guides. The other thing to keep in mind is that when you're preparing either plasmid DNA or in vitro transcribed single guide RNA, this takes several different steps. It takes quite a bit of time, and because each time you prepare it, there might be some variability in your preparations, this can result in, first of all, a labor-intensive process, but also variable editing efficiency. And so because synthetic modified guide RNAs can be purchased and they're very rigorously QC'd, They often exhibit very high editing efficiency, and it takes very little amount of time in order to prepare these reagents. So single guide RNAs come ready to use, and CRISPR tracer, if you get the two-part system, you need to anneal it within the lab, but that takes about 10 to 15 minutes compared to hours of trying to clone and purify plasmids or single guide RNAs that are in vitro transcribed. So at STEMCELL, as I mentioned, we offer both the two-part format as well as the single guide RNA format. So we wanted to test in human pluripotent stem cells whether there's any difference in performance between these two formats. So the strategy we used is using human pluripotent stem cells that are tagged with enhanced green fluorescent protein. So they contain a GFP transgene within their genome, and so they express GFP. And so this is what the GFP sequence looks like. And in these cells, we electroporated them with RNPs that contained Architect CRISPR or Architect Cas9, as well as Architect two-part CRISPR tracer RNA or single guide RNA. And these guide RNAs were targeting the GFP gene. And so the target sequence is shown above in orange right here. So interestingly, because GFP differs from blue fluorescent protein by only a couple nucleotides, we were also able to monitor knock-in efficiency by including a single-stranded oligodeoxynucleotide sequence that converted the GFP gene to BFP. So it contained the nucleotides that are required in order to convert GFP to BFP fluorescence. In our design, we also included some silent mutations in the SSODN to disrupt this protospacer sequence with additional nucleotides and to prevent guide RNA reannealing and recutting and the potential formation of indels to help increase knock-in efficiency in this system. So as I mentioned, we electroporated RNPs containing either the two-part or single guide with this SSODN. We then cultured those stem cells, and this enabled us quantitative assessment of editing efficiency by flow cytometry. And so we can monitor both knockout efficiency by looking at the percentage of GFP negative cells, or knock-in efficiency by looking at the percentage of BFP positive cells. And so this is an example of what the data looked like. In mock electroporated cells, they highly express GFP. When you only introduce the RNP that contains a guide RNA targeting GFP, you see indels or loss of GFP expression in a proportion of these cells. And then when you introduce the RNP with SSODN, you can observe some knockout cells as well as some knock-in cells. So comparing the two different formats, which was the ultimate goal of this experiment, we found that single guide RNA format outperforms the two-part format in terms of both knockout and knock-in efficiency. And this was significant, especially with the knockout efficiency measurement. These are example flow plots of the data that we gathered, and there was a trend towards an increase in knock-in efficiency with this single guide RNA format. We also wanted to verify this at a second locus, and so we chose to target the B2M gene, or beta-2 microglobulin, which is a gene that's required for surface expression of MHC class I molecules. And so when you target this gene, any knockouts will result in a loss of MHC class I expression on the cell surface, and so we were able to detect this using flow cytometry, and here are some example flow plots from our data. And again, what we found was that single guide RNAs outperformed the two-part system in terms of knockout efficiency as well. And so our conclusion is that, at least at these two loci and likely at other loci as well, single guide RNAs exhibit enhanced activity in human pluripotent stem cells. And now we understand that it can be challenging in order to design your guide RNA sequence. And so recently we also launched a guide RNA design tool. And so this allows you to either design your own custom sequence and input it here, or you can search pre-designed by gene. And so our tool uses the best practices and latest computational tools to deliver an optimal guide RNA sequence to knock out any gene in the human or mouse genome. And so you can enter your gene name here and choose your genome and then search, and at least 5 guide RNAs will come up that exhibit high activity and low off-target effects. or theoretically high activity and low off-target effects. We do suggest that at least 3 to 4 of these guide RNAs need to be tested because the computational tools are only so good. You also need to empirically test these guide RNAs, but it gives you a jumping off point to look, or I guess to design your guide RNAs for knockout. Okay, so that brings me to the next challenge, which is that human pluripotent stem cells are inherently more challenging to culture and manipulate than immortalized cell lines. So one of the main aspects of this challenge lies in the fact that HPSCs actually require daily feeds. And I know when I'm in the lab, I don't love that aspect of HPSCs. They're a really exciting type of cell to culture, but can be very finicky and very needy in culture. And so this can result in a lot of time spent changing media when you really just want to get to your experiment and get to those exciting results. And so in order to overcome this challenge, last year we launched a new HPSC maintenance media that's known as mTeaser Plus. And this is based on the mTeaser formulation, but we've improved it in 2 different aspects. So one of the reasons you need to feed HPSCs daily is because some of the key pluripotency factors are not very stable at 37 degrees, and this includes FGF2. And so the media needs to be changed daily so that FGF2 levels can be replenished. Another reason is that PSCs have quite a high metabolic rate, and this can result in acidification of the medium and an unfavorable environment for their culture. And so this also results in you having to change the media daily. So these are the 2 features that we tried to improve in mTeaser Plus, which is stabilized FGF2 as well as enhanced buffering capacity. And so the data is shown here. When in mTeaser 1, if you monitor FGF2 levels over time, you can see that by about 24 hours, the level of FGF2 is greatly reduced in the culture. Whereas with mTeaser Plus, you can see that FGF2 levels are maintained up to even 72 hours after changing the media. Furthermore, this data shows the pH level after 72 hours with increasing number of cells, and we compared mTeaser 1, mTeaser Plus, as well as an alternate HPSC maintenance media. And you can see that mTeaser 1 and this alternate media exhibit a lower pH, or so a more acidic medium, after this time point compared to mTeaser Plus, which shows enhanced buffering. Okay, so to just, I guess, compare or ensure that these changes that we made to the mTeaser 1 formulation had no impact on editing efficiency, we again used the GFP to BFP conversion assay to monitor editing efficiency in a quantitative way. And overall, we found that comparing mTeaser 1, mTeaser Plus, and this alternate HPSC maintenance medium, that there was no difference in editing efficiency across either 1C IPS cells that were tagged with eGFP or H1ES cells tagged with eGFP. And this was true for both knockout efficiency shown on the left, as well as knock-in efficiency shown on the right. One thing that we did note was that you can see a difference across different cell lines, and this was correlated with the growth rate of these different cells. So we know, because we've cultured them for a long time, that 1C cells grow quite a bit faster than H1 cells, and so we noted this relationship between growth rates and overall editing efficiency. And because of that, we do recommend that customers collect cells during their exponential growth phase when you want to edit those cells. And that editing efficiency will likely go down if the cells are overly confluent in the well, as those cells are likely arresting in the cell cycle. Okay, so this brings me to the next challenge, which is that HPSCs exhibit very low clonogenicity and single cell survival. So I'm going to bring it back a little bit to the PSC genome editing workflow. So we we design a guide RNA, form the complex, deliver it into the cells, and then plate those cells. And everything I've been showing you up until now is an analysis of this heterogeneous mix of edited cells. And so what we also refer to this as is a genome edited cell pool. And if you have high enough editing efficiency and you're looking to knock out a gene and study its function, this edited cell pool can be really useful for that. And it can be very similar to an RNAi knockdown experiment where you have loss of your gene of interest in a number of these cells. However, most of the time, and because of the ability of generating clonal cell lines with HPSCs, many people do want to generate clonal cell lines. But I would like to just note that if a clonal cell line is desired, this stage is not useless. This stage is actually quite useful in assessing editing efficiency and informing how many clones you need to screen in order to successfully derive your clone of interest. And so the way this is done is you can PCR amplify around your target region and send it for Sanger sequencing. And so you can compare the wild type sequence to your mixed pool, which will look something like this, which is just a jumbled mess. But there is this program known as TIDE, which is published in Nucleic Acid Research in 2014, that can decompose these sequence traces and can give you an idea of the relative proportion of alleles or of edited cell types within your population. And so in this example, they show that 34% of the cells remain wild type, 14% have a +1 insertion, 30% have a -1 insertion, and so on and so on. And so if you're looking to generate a clonal cell line, for instance, with a -1 deletion, that you know that you're going to have to select at least 1 out of 3 or 1 out of 4 cells in order to get your clone of interest. And so again, this kind of analysis first of all, has been shown to highly correlate with next generation sequencing. So it is quite representative of what's going on in the culture. And it can be informative, again, of how many clones you have to select for further characterization and can give you a ballpark idea as to whether how many plates you need to plate, et cetera. And so that brings us to the derivation of clonal cell lines, which at STEMCELL we often perform in a 96 well plate or 384 well plate format, where you can either plate your heterogeneous mixed genome edited pool at limiting dilutions to try and get only 1 cell per well, or you can use fluorescence activated cell sorting in order to sort cells into individual wells. And this will allow you to derive clonal HPSC lines. But as I mentioned, this stage is quite challenging because PSCs demonstrate very low cloning efficiencies and low single cell survival. So when you plate only 1 cell per well, the PSCs aren't very happy. They like to have neighbors, and so this often results in cell death or a failure of those cells to grow into a colony. Furthermore, genome editing is a very stressful process. You're incurring DNA damage, and that places a high degree of selective pressure on the PSCs. And so because of this, we do recommend to perform that editing step and the cloning step sequentially as opposed to at the same time. And this will just minimize the amount of selective pressure that you're putting on the cells at any given time. Okay, so in this process, a number of bottlenecks have been identified that are restricting single cell survival, and that's just shown on the right here. So this includes survival post-plating, so the cells need to adhere to the plate and survive that event. They then need to re-enter into the cell cycle and survive that after they divide. And so ultimately, this results in a less than 1% cloning efficiency for human pluripotent stem cells. And now there have been several small molecules described in the literature that help to improve single cell survival of HPSCs, such as ROCK or ROCK inhibitor. And this is often used to increase cloning efficiency, and this can increase it up to maybe 1 to 10% depending on your cell line. But when you're performing editing and you want to clone your edited pool, you really want to get higher levels of cloning efficiency than this. And so we launched a product known as Cloner that enables the robust generation of clonal HPSC lines, and it acts as a survival supplement that can be added into the mTeaser media, so either mTeaser 1 or mTeaser Plus. And compared to ROCK inhibitor on the left here, you can see that the Cloner induces a much higher cloning efficiency, and this is across a number of different cell lines. Furthermore, when you compare mTeaser 1 or mTeaser Plus supplemented with Cloner, the colonies that you observe with mTeaser Plus supplemented with Cloner grow quite a bit quicker and are much larger. And so this is also beneficial when you're trying to go through a genome editing workflow, as potentially you can start to select and characterize these clones sooner. Okay, so once you have derived your clonal PSC line, obviously it's not trivial, it's going to be a lot of work. You're going to want to characterize those lines and ensure that those cells remain pluripotent, that they contain your edit of interest, that they have minimal off-target effects, and the list goes on. And what I'd like to highlight now is what we do at STEMCELL when we're characterizing clonal cell lines. And there's a few different characteristics that we look for. So again, I'd just like to reiterate that editing is a stressful process. And so throughout this workflow, you're placing a high degree of selective pressure on PSCs. So it's very important once you've gone through all the work to ensure that cell quality is maintained after editing. And this is also important when you're just maintaining your PSCs in culture. And so at STEMCELL, I guess, different characteristics we look for are morphology, which is probably the easiest way to tell whether your cells are behaving properly. And so you want to have that characteristic PSC morphology of round colonies with smooth edges, high nucleus to cytoplasm ratio, multi-layering, etc. We also look for maintenance of the undifferentiated state by marker expression, and so that's shown in the middle panel here. So compared to a parental line in terms of TRA160, which is a protein that's highly expressed in cells that are not differentiated, we see that there is maintenance of expression of that marker in both non-edited or mock-edited cells, as well as our edited cell lines. The other, I guess, method to determine whether your cells maintain a certain level of quality or that they are still pluripotent is measuring whether they can differentiate into all three germ layers. And so traditionally, this is performed using the teratoma assay. And we recognize that that assay is quite challenging to accomplish and usually outside of the scope of what many labs want to do. And so we have, or we offer our STEMDiff trilineage differentiation kit, which has been, I guess, two-dimensional methods of differentiating cells or iPS cells in vitro have been shown to be useful as a surrogate for pluripotency or as a surrogate for teratoma formation. And so you can take your iPS cells and differentiate them using different media formulations into ectoderm, mesoderm, and endoderm, and this ensures that your cells are still able to differentiate into all three germ layers and still have that ability, or I guess that characteristic of pluripotency. So for the rest of the talk, what I'd like to focus on is the final measurement of cell quality that we are very interested in at STEMCELL, which is genetic stability. And so this is an example karyogram from a male cell line. And so this is what you would expect to see. And sometimes what you can find is when you send your cells for karyotyping is that they harbor some abnormalities. So in this case, this cell line harbored triplication of chromosome 12. And this is one of the most common abnormalities that can be observed in pluripotent stem cells during long-term culture. And we believe it's because chromosome 12 has genes on it that help confer a selective advantage to the cell population. And so we think This is especially important in the context of genome editing since you are introducing DNA damage and you are likely culturing these cells for a long period of time, that it's really important to continually monitor genetic stability. And this is because, let's say you've done years of work, you've generated clonal cell lines, you've gone through all of these different functional assays with your cell lines studying the specific mutation that you generated, and then you go to publish. Often papers will now request that you perform some sort of genetic analysis either by SNP array or karyogram. And if you get your data back and it shows that one of your cell lines or multiple cell lines have some sort of abnormality, this brings all of your data into question because you're not sure if you're studying that abnormality, whether that abnormality is synergizing with your mutation of interest, and it just becomes a big old mess. So we really want to try and prevent this for our customers. Okay, and this is very highly related to the final challenge that I'm going to describe, which is that PSCs are very sensitive to DNA double-stranded breaks, and that the damage response can constrain editing efficiency. Okay, so during routine culture, cells are under constant selective pressure to survive and maintain self-renewal at the expense or avoiding differentiation and apoptosis. So when you genome edit these cells, you are incurring DNA damage in these cells, and because HPSCs in particular have robust surveillance mechanisms and very low threshold for DNA damage, they will often choose to undergo apoptosis at the expense, or opposed to undergoing the risk of self-renewal in the context of a damaged genome. And so there's one molecule that really sits at the center of these decisions, and that's p53. And unsurprisingly, in 2018, two groups reported on the phenomena that the p53 pathway is activated by CRISPR-Cas9-induced double-stranded breaks, and that this response reduces the efficacy of genome editing. So importantly, this indicates that p53 status should be carefully monitored before and especially after editing. And this is also highlighted by this paper, which came out a year prior, showing that just PSCs during normal routine maintenance can recurrently acquire and expand dominant negative p53 mutations. And so p53 is obviously a molecule, especially in PSCs, that helps provide a selective advantage to these cells. And so especially in the context of genome editing, this makes quite a bit of sense because if you have wild type cells that have functional p53, a double-stranded break induced by CRISPR-Cas9 will result in activation of p53 and its downstream effectors like p21. And then the possible outcomes of this can be either that no double-stranded break is formed because no CRISPR components got into those cells, efficient genome editing. However, there's also the chance that those cells will choose to either arrest in the cell cycle or undergo apoptosis, and you won't see efficient editing in those cells. And so overall, the number of cells that have efficient editing is lower in your population. And this is in contrast to when you have non-functional p53, where p53 will not become inactivated, so you will not have those cell cycle arrest or apoptosis options, and you'll see more cells that have efficient genome editing. And so while on paper this sounds great, you'll get more cells that have your edit of interest, again, you will be studying cells that also are non-functional for p53, and you won't be able to tease apart whether your mutation of interest is causing a given phenotype that you're observing, or whether it's also because you have loss of p53 in those cells. Furthermore, if you ever want to take these cells to the clinic, it's going to be nearly impossible because p53 is a tumor suppressor, and so mutations in this gene do cause the cells to become, or can cause them to become tumorigenic. So besides, and again, all of this just kind of highlights the fact that while you're culturing your cells and before and after editing, you're going to want to monitor p53 status. Something else that is really important to monitor are some of the other recurrent genetic abnormalities that I mentioned previously. And so these are chromosomal abnormalities that occur during long-term culture of PSCs, and they can be selected for. So in this paper from 2016, they showed this ideogram, and they performed analysis of the most common abnormalities that were found in PSCs during long-term culture, and this included gain of whole chromosomes or chromosome segments, losses, shown here in red, or rearrangements. And you can see chromosome 12, which is one that I highlighted earlier, is one of the most frequently gained frequent whole chromosome gains in PSCs. And there's some other common ones like chromosome 17 gains and chromosome 18 losses. One that I'd like to highlight that's especially relevant to genome editing is chromosome 20. Chromosome 20q11.21, which is this minimal region shown here, is a very frequently gained region during long-term culture in pluripotent stem cells, and it helps to provide a selective advantage to these cells. And so in this study, they identified that within that region, it's really the BCL-XL gene that helps to confer the survival advantage. And interestingly, BCL-XL actually lies within the p53 pathway. And so it's downstream of p53 and the BH3 only proteins, and it's negatively regulated by p53. BCL-XL itself is a negative regulator of the pro-apoptotic molecules BAX and BAC. And so loss of, or overexpression, of BCL-XL, either through amplification of 20q or through other means, can result in inhibition of apoptosis, and so thus providing a selective advantage to those cells. And this is relevant to editing because in 2018, this group showed that when you transiently overexpress BCL-XL, that you get higher cell survival after editing, as well as much higher editing levels compared to without BCL-XL overexpression. And again, while this may be beneficial in the short term, and especially if you can control BCL-XL expression to be only very transient during the genome editing process, this could be a good thing. However, if you have amplification of 20q, again, you might be getting high editing efficiencies, but you don't know how to tease apart whether you're studying your mutation or the consequence of 20q amplification. So to address that concern of recurrent genetic abnormalities, we launched a qPCR-based genetic analysis kit, and this contains primer probe sets that cover 75% of the most recurrent culture-dependent abnormalities, including 20q. And so 20q we find is an especially interesting component of this, because when we do identify 20q amplification in cell lines, when we send those cells for karyotyping, often you cannot identify that those cells are abnormal because the region on 20q is so small. But this was validated by FISH, and so this indicates that potentially this kit can be used specifically to identify this 20q abnormality, which can be providing a selective advantage to your cells. And we feel like this whole kit, because it's quite a bit cheaper than karyotyping analysis, and it's also much more straightforward and in terms of analysis and the workflow, and you can perform it much quicker, this kit can be used to pre-screen your edited lines to ensure that they don't exhibit these common karyotypic abnormalities before either sending for karyotyping or performing any downstream analysis of those lines. Okay, so that brings me just to a summary of the presentation. So I went through some challenges and how we're trying to address them at STEMCELL Technologies. The first main point was that the RNP system, especially using synthetic single guide RNA, is the best available method for genome editing of human pluripotent stem cells, and we feel that this is the most efficient method with the lowest chance of off-target effects. And then the rest of the presentation was really focusing on pre and post editing culture conditions and the considerations to take into account for high efficiency editing and high efficiency cloning. And so some recommendations that we have are to edit the cells during their exponential growth phase, to use robust media systems that support post-editing survival and clonal derivation, to perform the genome editing and clonal derivation steps sequentially to minimize selective pressure on those cells. And we really just want to highlight that it's very important to monitor genetic stability during long-term culture, and especially after genome editing these cells, because all genome editing involves the formation of DNA damage. So with that, I'd like to thank some people at STEMCELL, part of the Genome Editing and Molecular Tools group, including Danielle and Jen, and the Pluripotent Stem Cell group, Adam, Matt, and Arwen, who have been really helpful in supporting this work. So with that, thank you for your time.